Viruses take selfishness to the next level. Viruses are selfish genetic elements that deliberately hijack the molecular machinery of a host to manufacture a phenotype. The phenotype improves the virus's ability to infect new hosts. Viral particles consist solely of genetic material, that is, a stretch of DNA or RNA, safely tucked inside a protein coat. They have no metabolism of their own. They rely entirely on that of their host. Here we see what are called bacteriophages. These are viruses that exclusively infect bacteria. And what's happened here is that one single virus will have infected the bacteria, gotten the bacteria to produce dozens of copies of the virus, and then they kill the host as they leave it. And so you see the viral coats attached to the surface of the bacterial cell wall on their way out. This animation shows how a virus attacks and invades a bacteria. Here's our cell wall of the bacteria with its circular chromosome. A virus is looking for a place to attach onto the cell wall, finds a receptor, then injects its genetic material into the cytoplasm of the bacteria. The viral DNA in this case is integrating into the host chromosome and becomes part of that circular chromosome. The bacteria may divide several times, and the viral DNA is still there. At some stage, though, the viral DNA separates out from the host chromosome, uses the host's machinery now to actually obliterate the bacterial chromosome, replicate its own DNA, manufacture more and more copies of itself, and then manufacture the protein coat. The virus then reassembles, breaks out, and goes off to infect more bacteria. So it goes in, it uses the host's own machinery to destroy its own chromosome, replicate the viral DNA instead, translate proteins into making a viral coat, supervise the reassembly, and then the exit from the dead bacteria. Now, bacteria as organisms fight viral infection. Viruses have probably been around infecting bacteria for billions of years, and so they have a lot of defenses to protect against these invaders. And one of the most important ones are called restriction enzymes. And these look for certain sequences that are more likely to be found in the DNA of the virus than in their own and then they cut it. So the restriction enzymes attack specific sequences that are mostly found in viral DNA and break it up. So in the last case, we saw a successful viral infection that broke up the host's chromosome. Here the host is actually breaking up the viral chromosome to prevent it from causing any damage. But these restriction enzymes work by finding a certain sequence that is very common in viral DNA and chopping it. Now with humans, we're also infected with viruses, and we have a far more elaborate defense system against viral infection. So what we're going to do is we're going to watch a virus come in and be detected by what's called a macrophage. And the macrophage then is going to initiate a number of processes that are going to disarm the virus and prevent it from doing damage. So the first thing is that the virus infects the body. The macrophages detect the presence of the virus, and if they can, they'll engulf it, and they'll actually swallow up at a cellular level the virus, and then they display some of the proteins that are found in the viral coat on their own surface. So these are called viral antigens. So these are now on the surface of the macrophage, and once this is started, there are various helper T cells that now know that they need to go and do something because there's a macrophage that has captured a virus. So the helper T cells activate what's called a cytotoxic T cell and also B cell. And by the way, you don't need to know all these different names. What I want you to get as we go through this is just how comprehensive the defense mechanism is within our bodies when we're infected by viruses. So the B cells form plasma cells. So these are actually very active in producing yet another kind of cell called a plasma cell. And the plasma cells make antibodies. These are now proteins that are now ready to go out into the bloodstream, and they will start looking directly for viruses themselves, and they'll bind on to the viral antigen. The antibodies now attach directly to the viruses, and they also look for infected cells that have somehow had the virus that's going in there and doing mischief. And they say, okay, this has got problems, this particular cell, and we need to have this cell destroyed before the virus can replicate itself inside the body. And so then these cytotoxic T cells that have already been activated by the macrophage, they're standing around waiting to find another cell, a human body cell, that has the antibodies on the surface saying, okay, this is where I go, and I'm going to break this cell down and destroy it. So an incredibly effective system that identifies these viruses, turns on in various ways mechanisms now to destroy the virus, and also the cells in our own bodies that it's too late. They've already been infected. Well, those have to be destroyed before the virus causes more harm. A remarkable aspect of our immune system is the way that the B cells are able to cope with almost any possible invader. So the B cells undergo extensive mitosis and form trillions of different variants. And each one of these is slightly different and can each specifically recognize a certain kind of protein. Now, the first thing in development is that those B cells that attack cells of our own body, that is, that would attach to self, are identified and destroyed. So now all we have is a system that's looking for variants of genes that are found outside of our body. So they're looking for aliens all the time. And virtually all possible aliens are being anticipated by our immune system. Now, when a foreigner does show up inside our bodies, the antigen is recognized by its counterpart B cell, because this round thing has a round receptor right here, and it says, okay, I'm ready to get going. And it proliferates. Only this one proliferates. And it produces what are called plasma cells. And the plasma cells are very important because they're the ones that make the antibodies to remove the antigens. So you have a specific response to the immediate threat of an invasive particle by the production of plasma cells uniquely suited to recognize and fight that particular target. But another thing that happens in this immune response is the formation of memory cells. And the memory cells are not going to be used up right now. They're dormant. They go to sleep. And they're there so that should at another time in our life we're exposed to the same virus again, they're ready to go. So we have an immediate response from the immune response, and we have a long-term memory waiting for that virus should it ever come back in our lifetime. Now, viruses ultimately arise independently a lot of different times. So if we ask where on the evolutionary tree viruses might exist with respect to other organisms, we see there's no one place to find the viruses. The bacteria are the oldest living things. They were followed by slightly more complex single-celled organisms and then multicellular organisms. Each has its own discrete branch on the family tree of life. But viruses are polyphyletic, that is, they originated over and over again. And what we see then is that if you sequence the genes of a virus, it turns out that a fungus virus, that is a virus that only infects fungus, has more genes in common with the fungus itself than it does with a virus that might infect animals. So these are the result of transposons that gave rise to viruses independently many different times. And when we classify them, we would classify their host, so they're fungal viruses, they're plant viruses, they're bacterial viruses, and they're animal viruses, or by their genome. Some viruses have inside the viral coat a stretch of DNA. Others, instead of DNA, they have RNA. So we have a class of viruses that are not evolutionary through common ancestry, but they've each independently arisen having RNA as their mechanism of inheritance. So viruses have this repeated origins because they have come from transposons. A transposon in plants became a virus of plants. A transposon in animals became a virus of animals. So we had initially just these simple transposons that could replicate themselves, get themselves from one part of the genome within the same host as another, whether they're plasmids or the jumping genes like Barbara McClintock found. But then we have a virus, which is essentially just a transposon. All it does is a virus is able to get itself somewhere else in a genome, but it has a phenotype. And that phenotype allows it to exist outside of a host for a prolonged period of time, in some cases weeks or even months, while it waits until it finds a new host. So viruses arise from selfish genetic elements that develop phenotypes to assist their movement from one host to another.